So test Tuesday, bring Scantron, 882E, number 2 pencil. You'll write on the test, unlike my lab class. You will write on the test, and you will write on your Scantron. And it's okay. I know. Tuesday. It's Tuesday. And so sometime very soon, today or tomorrow, I'm going to pare down the list of predict the outcomes from the crosses that you guys will have to do for the test, right? All the rest of the objectives you should be able to do, define, distinguish, compare, contrast, the different types of dominance and all of that.
Will you do some chapter 5 stuff on Connect? Or actual study?
Yeah, I'm going to put some chapter 5 stuff as well. Yeah, because those problems, hopefully they have some good problems. I'll try and do that for you guys tonight. Okay, so we got to finish up chapter 5 first, and then nobody sent me any problems, but Christine sent me a link, which was hysterical, perfect timing for this chapter, right? So I'll add that for you guys. I did read it, and the hysterical thing is that a colleague, Christopher LeBlanc, actually was outside yesterday smoking, and I had to talk to another friend who was smoking, so I had to inhale some secondhand smoke. But he's like, "Did you hear about the three DNA baby or whatever?" And I was like, "Yeah, a student sent me the link, but I haven't opened it yet." I was like, "Give me the skinny on it," and I was like, "Oh, okay." So it's not 3 chromosomes. It's 3 different things of DNA, which is what we're going to be talking about today, about how your mitochondria, remember, have their own separate DNA. I kind of prefaced this earlier. So there's actually been a case, right, a real life case, where they used the mitochondria from one parent, the DNA from a different parent to form the egg, right? So they took a donor egg, right, and they took out the donor egg's chromosomal nuclear DNA, right? So just left behind all the organelles, which, of course, the mitochondria, as we know, has its own DNA. And the reason why they did this is the poor mom had several babies she had lost to a genetic defect in her mitochondria. So she couldn't have babies of her own with her own eggs because her mitochondria were defective. So they took a donor egg, they took out that donor's DNA and put in this woman's DNA, right, her chromosomal DNA, and then fertilized it with her husband's sperm. And then they took that embryo and implanted it into her. And the baby has been born, it's 6 months old, it's fine. But it's controversial, right? Because we're basically playing God. Wow, that's great. I don't doubt it. Yeah, it is creepy.
But that woman in Texas who was in the coma, they said that, even if you're in a coma and you have a baby inside you and you're pregnant, the baby doesn't do well because it needs you to be alive.
Right.
So, they just said, so there's actually a bill in Louisiana that if you get in an accident and you're in a coma and you're pregnant, you have to stay on life support.
So that the baby can be born, yeah.
Period. Yeah, no matter what.
Yeah.
Which is pretty awful.
Yeah.
But scientists and doctors say that the babies don't do well
because of you need all the other stuff to happen. Yeah.
The whole person. Not
yeah, my child probably thinks that all I did was yell because I was a teacher when I was pregnant with him. So yeah. He's probably used to the loud voice. But yeah, no, I mean, there's so many studies that have been proven that prove things like that, you need all of that stuff that happens, that when the mom is alive for that baby to develop normally, to have normal developmental. So, yeah. Okay. But that's definitely, especially in genetics, as we talk about these things, there's a huge ethical component, what is or is not ethical, you know, where are we as a nation, as a country, as a state going to draw certain lines as it relates, and how are we going to determine if we're going to cross that line for certain circumstances. So a lot of different things to definitely take into consideration. And it's not, and the thing is, we draw these lines, but is life really black or white? It's really not, you know. It's a whole lot of grays. So I don't want to spend too much time on that, but I will post that link that Christine sent me to that article that talks about what they did. And of course, they didn't do it here. They did it in Mexico, where they don't have any laws against it. In the United States, they have laws currently against what they did. So it's not allowed. So they went elsewhere so they could have a baby. And they specifically did a boy. Why did they do that? So that donor mitochondria will not be passed on to the next generation. Because he's a boy, right? He doesn't make eggs. He's going to make sperm. So his children will only get a combination of his parents' DNA. That mitochondrial DNA that came from the donor is just going to be with him. It's not going to be ever given to anyone else. So it just made him possible. Pretty crazy. All right, so, That's what I'm saying. It's not 3 different DNAs per se, right? It's the parents, the chromosomes, right, are from his parents. The only DNA that's not from his parents is what's in his mitochondria, because his mitochondria came from the donor. But the reason why they did that is because his mom's mitochondria have a serious defect, so much so that all her children were dying. Right. Yeah, they actually list what particular disease it was. No, she has enough, and we're going to talk about this today. She has enough normal mitochondria for herself, but when they were getting divided up to make her baby, she kept giving too many bad mitochondria. They weren't winning the genetic lottery. Right. So, last time, I think, did we end basically getting ready to start talking about the extra nuclear. I know I didn't. Oh, methylation. Okay, I did. We did. Yeah. So I was talking about how at the molecular level they shut down one of the X's, and then we talked about the different size, the imprinting in the mice, but we didn't talk about it at the molecular level, right? So I'm going to skip ahead to not that slide. Where is it? Ones with the little thingies. There we go. Okay. So how is it, and we kind of saw this with the forming the bar body, how does the cell know that it's not going to use that particular chromosome, right? How does it know it's not going to use that information? It modifies it, right? You guys saw that even in the animation, that video that I played on the forming of the bar bodies. So they add methyl groups to the DNA and even to the histones, the proteins that complex with the DNA. So the enzymes know this is the one we're not using, right? It's been methylated. It's basically been tagged that this is the one we're not using. Okay? So at the molecular chemical level, they turn off basically that chromosome. They don't use that information. So with imprinting, it's, this is the same thing, except it's not a complete shutdown, right? It might be just a region. But again, they're not using that information. So for when the gametes are forming, right? They erase the imprinting that affected them, and then they're going to re-imprint depending on that pattern of imprinting for that trait. So in the case of this one that we looked at, was this must have been maternal, right? So the males, right, they methylate their DNA, and the females do not. So I'm trying to remember this example. Notes, genes, such a headache today. Normal size, dwarf size. Oh, here we go. So males in this case are actually transcribed. So is this a different one that they give us the example for? Yeah, methylation causes inhibition of the transcription. So this must be some other trait that they're doing, because the males don't methylate for the dwarfism. It's the opposite. Why did they do that? They didn't give us the same example. It's kind of annoying. The important thing is for you guys, right, is that they're going to chemically modify. One example is methylation, that's used a lot, to modify the DNA, to basically tag it so that the cell knows this is not what we're using, right? We're not going to use this information, we're going to use the other one, right? So just like with the bar body, it gets methylated, but it also gets condensed. There's even additional proteins in those RNAs that really condense down that extra X that we have so that we don't use it. We only use the one that doesn't get shut down. And then for us, for mammals, right, it's random as to which X gets shut down, right? It isn't predetermined whether we're going to shut down the one we got from our dad or our mom. Whereas some of the other ones, right, there's a particular pattern as to which one gets shut down, or if they upregulate instead. By no means do I want you guys to memorize that table, right? I don't have it memorized, right? These are types of things you can look up. A general understanding of how these things work is what we need, right? That these things happen, and why do they happen? Well, they happen because we've got to compensate somehow for the fact that one sex has 2 chromosomes that are the same, and the other sex only has 1, right? Do we upregulate, right? That one that has just 1, do we make more from that one? Do we downregulate? Or in the case for us, do we shut one of them down, right? How do we compensate for these differences? So the example they give for another example of imprinting is linked to a particular genetic disease. And again, these are just some examples, right? And sometimes it helps us understand it when we have a concrete, real-life example, right, to hold on to. So the one for this one, for the human example as opposed to the mice, is an interesting phenomenon where in one sex you end up with the PW syndrome, abbreviated PWS, or Angelman syndrome, abbreviated AS. And it's 2 different genes, or I think for one of them they're not exactly sure what exact genes are involved, but it's definitely a difference in imprinting between the different sexes, between males and females. So for this particular one, the PWS, this particular syndrome, the individuals have decreased motor function, obesity, small hands and feet, where AS is they're hyperactive, they're really thin, unusual seizures they'll experience, and repetitive, systematic muscle movements, and as well as mental deficiencies. So neither one, right, is good. They believe that they have to do with a small deletion that's in chromosome number 15. But why we see two different outcomes from this is because imprinting differs between the male and female sex as to which one is allowed to be active or inactive. So if it is inherited from the mother, they have AS. If they inherit it from the dad, they have PWS. So why is that? So again, it's a problem, it's a defect in a gene, right? They're not able to do something they need to do to be normal. So they have these defects. So that particular gene that leads to the problem, the paternal copy is silenced. Where with PWS, it's a maternal silencing. So let's look at an example. So the two genes are on the chromosome 15 that need to be working and functional. And say we have another gene that it's completely deleted, right? There's nothing there to make the gene products that you need to make. So the chance for the mom for her eggs is not have anything at all, or have the defects. But the thing is that she is going to silence PWS, right? She's not going to silence a defect she may have in the region that is going to cause AS. So remember, she imprints her eggs. Males, on the other hand, the sperm are going to be imprinted in the opposite way. So in this case, the male sperm, right, it has turned on PWS and turned off AS. So when this sperm combines with this egg, that embryo, that baby or whatever, is going to have PWS, because that's the one turned on because it came from the sperm. It's not going to have AS. Where if it's the mom's chromosome with the defects, mom turns on or allows AS to be accessible, but PWS is shut down. This sperm didn't contribute any of these, so it is the mom that determines this zygote is now going to have AS as opposed to PWS because of the mom's imprinting, right? What mom turned on or off. Now, like I said, the exact right down to the molecular why one's turned off versus the other, I haven't quite figured that out yet. But you know, this is another case of imprinting. What are you turning on and what are you turning off? And that's why in the different sexes, depending on where that came from, will determine what disease you have. So the last one is what we've been kind of talking about, the fact that mitochondria and chloroplasts have their own DNA. And part of the reason for this, we believe, it has to do with the endosymbiotic theory. And that is the theory that these organelles were once free living organisms living all on their own, right? And they were engulfed by another cell, a pre-eukaryotic cell. And they began this symbiotic relationship. So much so that it got to a point where the chloroplast or the mitochondria lost some of their information, and some of that information may even have migrated to the nucleus. Because we know nowadays when you look at the DNA, there is DNA that copies stuff in our nucleus to make proteins and such for our mitochondria. But some of the things that the mitochondria need, they make right inside the mitochondria from their DNA using their ribosomes. They make their own ribosomes, right? They make their own transfer RNAs. They synthesize some of their own proteins right in the mitochondria. But they can't do enough on their own, right? They can't go and live on their own right now, because they don't do it all, right? Some of it is done by the nucleus of the cell. But not all of it, right? So it's that knowledge that we have, that we know that they have DNA, they use their own DNA. And that structure of the DNA, the structure and sequence of those ribosomes is very reminiscent of prokaryotic cells. Their ribosomes are actually the size of prokaryotic ribosomes and match sequences similar to bacteria. So they actually believe that the modern day ancestors of the mitochondria are actually cyanobacteria. Bacteria that basically are like a chloroplast, but on their own, right? They have everything they need. They can do exactly what our chloroplasts do, but they live as single-celled organisms all on their own. And then there's, the I don't want to say the name wrong. I'm going to flip to it. Hopefully it's in here. What are our mitochondria? Ah, there. I think it's non-sulfur purple bacteria. They didn't put the non-sulfur part. So there's a group of bacteria out there that use sulfur as electron donors. And most of them are purple, but not all of them. That's where they got that name, purple bacteria, because they literally are purple. Some of them because of the pigments that they produce. So those modern day bacteria have very close similarities to our mitochondria. Right? So our mitochondria, again, just like chloroplasts, have prokaryotic ribosomes. They have DNA to make those ribosomes, make the transfer RNAs necessary, and they make some of their own proteins that go into the membrane to form the electron transport chain to allow that mitochondria to make ATP. So again, that created a problem for this woman because her mitochondria were defective. And so much so that it caused the death of her children. They couldn't do what they needed to do. Does that make sense to you guys? It's kind of fascinating, cool stuff, huh? To think that, it was a symbiotic relationship that became so dependent on each other. You can't divide them anymore, right? We can't survive without our mitochondria, and they can't survive on their own, right? So where did I want to go? I kind of jumped ahead. So again, this might be one of the predicts I give you guys, probably, maybe not, I don't know. I gotta finish my list. But you should definitely be able to explain how mutations, right? Whenever you go to any of these lists of objectives for each of the things that we've been talking about, you need to be able to describe the general features of mitochondria and chloroplasts. It's when you get to predict the outcomes of crosses that I'm not going to make you guys do them all, and I'm going to pare down the list to make it easier for you guys as you're preparing for this test on Tuesday. But you should be able to explain how the mutations in the mitochondria cause human diseases and what we mean by endosymbiotic theory. So we already know, right, that the chloroplasts and mitochondria, they have their own DNA. And so they kind of do their own stuff. So just like bacteria, that region where the DNA is, it's called the nucleoid. And it's circular A single double-stranded DNA. So again, very much like what prokaryotes bacteria have even today. But the interesting thing, though, about these organelles is that they tend to keep several copies of their chromosome. Where prokaryotes bacteria, when they're only usually copy their DNA when they're getting ready to divide. They don't usually keep extra copies of their DNA. But for some reason, these organelles do, and it really varies a lot between species and different types of organisms. So just as an example, you'll see in mice, in the mitochondria, they'll have 1 to 3 nucleoid regions. That's a region where the DNA and the protein that complexes with it are located. Within each of those 2 to 3 regions, they will keep 2 to 6 copies of that circular chromosome. Where you've got someone like Euglena, as far as their chloroplast goes, within the chloroplast, they will have 20 to 30 regions where the chromosome is. And within those regions, they'll have 10 to 15 copies of their chromosome. It's a crazy lot of DNA. Right? Why do they do that? You could surmise lots of different reasons why, but the fact is they do, right? They do this. And so, as you can see, it varies quite a bit. And so for the mitochondria, the function is oxidative phosphorylation, right? Using the electron transport chain, move electrons along, create that gradient, move it back through ATP synthase, and generate ATP. So it doesn't mean oxidative in that you're using oxygen, right? It means that you're oxidating, you're moving electrons, right? Because some organisms are not like us, they do it differently. But for the most part, right, we're using oxygen as the final electron acceptor, right? It's accepting those electrons at the end of the electron transport chain, and we're generating ATP. So we commonly abbreviate it when we refer to the DNA in the mitochondria as MT for mitochondrial DNA. So our mitochondria DNA is relatively small chromosome, 17,000 base pairs, right? Where for our chromosomes, right, we're talking about like 4 billion base pairs. So this is like teeny tiny in comparison. And so it doesn't have a lot of information, but it has information it needs, right? And it needs to function. So it has, as I said, the ability to make its own ribosomal RNA, to make its own ribosomes, and it has the transfer RNAs it needs to make proteins, right there. And 13 of the proteins that it needs to be able to do the electron transport chain, right, oxidative phosphorylation, it makes itself, right there in-house. But most of all the others that it needs are in the nucleus of our cells. So they've got to be made into messenger RNA, travel out of the nucleus into the cytoplasm, be made into proteins, and then transported into the mitochondria. So here is a map of the circular chromosome, and you can see, as we said, lots of transfer RNAs for all the different amino acids. And then where is it? There's one part of the ribosomal RNA, and there's the other part of the ribosomal RNA that make up the subunits of the RNA. And then the rest of these bigger sections, right, are different proteins that are needed in the electron transport chain. Like cytochrome B is this pink section. Cytochrome C, oxidase subunit genes, there are 3 of them in the light blue. I got pink on the brain. Right? They actually have a section here that doesn't code for anything. Interesting. Right? So they can do some things on their own. The problem is they can't do it all, right? And the problem for that woman is that her DNA for her mitochondria didn't have everything it needed. Right? And the nucleus didn't have it either. So for chloroplasts, we know that's photosynthesis, right? And it's not all that different from mitochondria, except the fact that the energy is coming from light. They're still shuttling electrons down a membrane, an electron transport chain, right? They're generating ATP. But again, they've got to generate some of their own stuff in-house. So they have their own DNA, and we denote their DNA as CP for chloroplasts. These guys are much larger, though, than mitochondrial DNA, about 10 times more DNA in a chloroplast. So for an example, the tobacco plant has about 156,000 base pairs, as opposed to, the mitochondria that only has 17,000. So it has a lot more genes then, of course, because it has more DNA. So it has anywhere from 110 to about 120 different genes. But again, they're making their own ribosomes, and they have their own transfer RNAs. So in-house, they're making their own proteins, just not all of them, right? They've got to get some of them actually from the nucleus of that cell. And they've got to be transported there. So maternal inheritance for most organisms is where you get your chloroplasts, your mitochondria. So it is the mom or the egg, right, that houses these organelles. The sperm or the pollen granule doesn't have any organelles. Or if it does, it's so few it doesn't factor in. So for the 4 o'clock plants, we talked about, they have that pattern of inheritance of incomplete dominance where you get the pink instead of the red flowers when you're a heterozygote. So when you look at their leaves, though, and their leaves are green based on the type of chloroplast they have and what pigments they have. And of course, the pigments are determined by what DNA they have and what they make, right? What proteins they actually make in that chloroplast. So some of the leaves have enough chloroplasts that produce the green pigment that they're green. Others don't produce the green pigment at all, and so they appear white. They still have chloroplasts, they just don't have the green pigment. And then other plants have both. They have both the white and the green variety. So depending on how many chloroplasts they have of the white or green variety in that cell will determine whether that cell appears to be white or green. So you get this variegated pattern, which is very common to what we talked about with the calico cat, right? Except the difference there, it's which X chromosome. Here, it's what chloroplast do you have, right, in your cell. And so you end up with these plants, that have both green and white. They're going to be what's called variegated. So those plants that have like the stripes, we have a picture coming up. So it's maternal. It's whatever the mom has is going to determine what the offspring look like. So here's mom plant, right? And she's white. She doesn't have any chloroplasts that have the green pigment. She's completely white, pure breeding white. The male, on the other hand, is got plenty of green chloroplasts, so he's all green. When these two mate, though, all of the offspring are white. Why are they all white? She's white. And where do they get their chloroplast? From their mom. So that's all she had to give was white, right? So that's all her children will be. because that pollen granule does not contain any chloroplast. It's just DNA for the most part. Still with me? So if you have a variegated mom, though, she has both white and green, right? So her children, depending on which ones get packaged up in that egg, they could be all green, they could be all white, or they could be variegated, where they have both green and white like her. Just depends on what she gives them. How does it get divided up when she makes that egg? And so the same thing, while you see this pattern, again, it depends on what each one of those cells ends up with. If you have the choice between both, you could be one or the other, right? Depending on what ends up in that cell. And that's the interesting thing about chloroplasts and mitochondria, is that, okay, they're made in the cell, and I think we have a diagram for this one that'll help us in understanding it. Yeah, so here's one. Okay. So in the green cells, right, when you just look at the leaf, if they've got some green, they're going to look green. But in reality, they could have some in there that are white. Or they could be all green. But when this cell right here, especially ones that have a mixture, when they go to divide, it's random, right? They don't put all the white ones in one cell and all the green ones in another one, right? You could end up, when this cell divides, she can end up, both the white ones go to one cell and both the green ones go to another cell. So now that's why you end up with these sections, because this is all they ended up with was with white, right? And all their offspring from that point forward are all going to just be white cells. But you're going to get the switch is when you have a green cell that has both white and green, because it's random. She may give one white and one green to each one of her cells. In that case, they're still going to be green, right? But at some point, she might only give white ones. And those cells and all those progeny are only going to be white. She might just give green, and therefore all the progeny are going to be green. But the fact that that organism started out with a mixture is why you end up with a mixture, this variegated pattern. Still with me? Okay. So this happens again because it's maternal effect, because mom is the one that has all the organelles. Dad doesn't contribute any. Dad just contributes DNA. But that's not always the case, right? But definitely for plants as it relates to chloroplasts, and for us as it relates to our mitochondria, that's how it happens. So here's some examples, of course, that deviate from that. So in the case of yeast, right, it's paternal inheritance. For the most part, for molds, it's maternal, right? So it comes from the mom. But there is one genus they found, oh, it comes from dad. It's paternal. But as I said, for us, right, it is definitely maternal. You get your mitochondria from just your mom. If you get any from your dad, it's so few, it doesn't factor in. It doesn't create a problem or help you out in the case of disease, right? So, and you can see for plants, right, there are different patterns as well. So the rare occasion where, say, dad's, mitochondria comes from the few that he has in his sperm to get that propeller to turn, to get to his mission, right? If a few of those actually get into the egg and get maintained, that's referred to as paternal leakage. So, a few leaked in, right? A few got in. But usually, as I said, it's not enough to matter. So when you're talking about, for instance, in a mouse, if you get 1 to 4 from your dad, but that's for every 100,000 you get from mom. It's so small, it's basically insignificant. So for us, right, mom makes all the organelles for us, makes this big, huge egg, right? Sperm comes along, fertilizes the egg. So we have this maternal pattern of inheritance for our mitochondria. So mitochondrial mutations, the problem with this is kind of like how we saw with the chloroplast. It really depends on what mitochondria end up in what cells. And for us, really, even what parts of our body. Whether you're going to end up having a problem based on what mom gave you. So, I'm trying to think if they have it here. Okay. So, mitochondria are very susceptible to DNA damage. And probably that's why they keep so many extra copies, unlike bacteria do, is in case one copy gets damaged, they get like a backup copy. Does that make sense to you guys? And it has to do with what happens in our mitochondria. Because oxygen and electrons are very reactive and they can damage DNA. So if you're producing a lot of this in the mitochondria, which you need to do, you need to produce a lot of energy. Occasionally, some of these electrons go where they're not supposed to, right? And the oxygen and crazy electrons attack the DNA. So because of that, like I said, it probably makes sense that they have so many extra copies so that, if one gets damaged, like, okay, I got to back up. No big deal, right? It's not the end of the world. I can survive. And they, unlike our nuclear DNA, don't have a lot of repair mechanisms. So if it gets damaged and they don't have those backup copies, then they're kind of screwed, right? They can't continue doing what they need to do. So there are about 200 human mitochondrial diseases that have been identified. So these are diseases distinctly linked to defects in the mitochondria. And so these tend to be chronic degenerative disorders because over time, right, as you have more and more of these mitochondria that are defective or became defective, the greater the problem is going to be. So I had a friend when I was in college that I met, and he was an older gentleman. He was probably mid-30s, maybe early 40s. He was going blind. And he had a disease that they mentioned in our book for this. He actually has one of the ones on the list. Which one is it? This one. The first one. Leber hereditary optic neuropathy. So the problem was the mitochondria he got from his mom were defective, right? Your eyes need a lot of energy, right? To power that optic nerve to be able to transduce that signal to know what you're seeing to the brain. So that takes a lot of energy. So you need a lot of very good active mitochondria in that region. If you don't have them, right, and as your cells divide, you end up with less and less effective mitochondria, then you can't do this as efficiently. So slowly over time, he was losing his eyesight because of those defective mitochondria. And in that particular area, it's very debilitating. So his sister, guess what? Chose not to have children once they found out about his disease and were able to figure out where it came from. Why is she not having any kids? Her mitochondria are defective, but she doesn't have the disease. The thing is that he had more mitochondria that were defective than maybe she does. And again, unless they go and check and test her mitochondria, she doesn't know how many of hers are bad. So she didn't want to run the chance of passing them on to her children and this happening to her children. So she has cats. She's like, I'll be a cat mommy. Right? You know, so she made that choice based on that information. Now, could she run down to Mexico now and have some babies?
She had a lot of money.
Yeah, she had a lot of money. Right? But I mean, unless she has her mitochondrial, all of it checked, right? All of it checked, right? To be sure that she doesn't have a lot of defective. Chances are she's going to pass that on to her children, right? So unless she gets mitochondria from a donor, right, her kids could have that disease. That could happen to them.
So could she wind up getting the disease later on in life?
Yeah, she could. I couldn't remember if she was older or younger than him. You know, these were details a long time ago I didn't think to ask. You know, but I was a science nerd, so I asked a lot of questions. You know.
But it did look kind of, depending on how many mitochondria are infected, it could strike it different.
Yeah,
Personally.
yeah.
Is that what you're all these? Or most of them?
Most of them, right? It just really depends on how many defective ones did you get from mom and where are they in your body even, right? Because as your cells divide, they don't do it any particular, there's no plan. It's random, right? Which mitochondria goes into which cell and how many of the good versus the bad. So that's why I think the plant one with the chloroplast gives you guys a pretty good representation because it's color related. But do you guys see that similarity there, right? So with that mitochondria, it's a mixed bag, right?
So like when babies form in the uterus,
Mm-hmm.
the mitochondria could get divided. Is that what you're saying? Like they're divided at that early stage, or
All throughout your life, right? So the thing is that it's random, right? So you're given this set of mitochondria from your mom. Say, for this poor woman that went to Mexico, she lost so many babies. I mean, I was almost crying reading that article. Miscarriages, one made it to like 6 years old and then died. Another one died at like 9 months or something like that. So, I mean, she had a lot of defective mitochondria because none of her babies survived, right? They all ended up dying. Granted, one of them lived to about 6 years old. But the problem is that, you know, not being able to do something depending on where it is in your body, you can only go so long not doing something where finally the body is done, right? It's done. In the case of my friends, in his eyes, you know, in the beginning, he had enough of the mitochondria there that functioned. But then it ended up when cells split, more of the non-functioning ones were in the new cells than functioning. Does that make sense? So now he basically has nothing but non-functioning mitochondria. So it's like having a whole bunch of white cells for that plant, right? When every time it divided, most of the time, even though there were some good ones, the new cells, they kept getting the old defective mitochondria. So then that's all you're left with, right, is bad mitochondria. So now you can't do it. You can't do it. You cannot do what the eyes need you to do to see.
but how it got to his eyes
Was again random.
Randomly when
Very early on in development, yeah. So you know, so again, you're an egg. Say, you know, he might have been like 50 of, you know, 5 of the mitochondria were good and the other 5 were crap, right? And when it divided, the ones that ended up be part of his eyes, he ended up with enough good ones that for a while he could see. But then it got to the point where in that random divide, he got all bad ones. All the new cells are not functional mitochondria. So he can't see. He's blind. It happened to him over the course of about 2 years. You know, all of a sudden he was having trouble seeing. Went to the doctor, they ran tests. They were able to pinpoint it back that it was this particular disease that he inherited from his mother. So he was learning Braille and, you know, learning as much as he could. I mean, and that probably wasn't any good either, because he was like, I think he was really like stressing his eyes, because I mean, like anything he could read that he wanted to read before he couldn't read anymore, he was reading. You know, every time I saw him, he had a book in his hand. I'm like, and you know, through the 2 years that I knew him in school, his glasses went from, you know, most people's glasses to, I mean, they were Coke bottle glasses by the time we graduated. He was almost completely blind in a span of 2 years. Pretty crazy. So what else do we need to talk about? Or do we want to try and get a couple problems in? Do you guys feel okay on this? Okay, all right. I'm not going to read verbatim. So nobody sent me, how did you guys, everyone hopefully at this point who's here today did their chapter 4 and Connect, and they feel comfortable with those? Those are pretty easy? Okay. Yeah. Oh, you did send me an email. Oh yeah, you did. Yeah, I know. I can look it up. Oh, some of you guys, I think, put it in the comment section. Well, I haven't graded them, so that's part of the reason, because I physically have to grade them. But I noticed that some of you guys, instead of like putting the text in or attaching a file, I think you put it in the comment section of the assignment, which is fine because I can see it. But I got a separate email and then I was like, oh, they put it in the wrong spot. That's okay, as long as it's there. As long as it's there, I can do something with it. Okay, so let me click over to my email, and Linnell did send me something, so let me search for that, because she can't remember it. Yeah, oh, so from the homework. Okay. Oh, okay. So, the pedigree chart. Okay.
Yeah. I got that one wrong too.
Okay. You know what?
It was true false. Yeah, it was only true false.
It was only true false? Okay. All right, so let's look at it. How do I get to that? We can flip through it real quick. So it is from the last Connect homework, though, right? Okay.
Yeah, it was the only true false one in there. Yeah.
Okay. Okay.
Yeah, I got that wrong too.
All right. So let's look at this. So the pedigree below shows inheritance of a dominant trait. Oh, polydactyly. We talked about this one, right? The extra fingers and toes. So individual 2-4 has 6 fingers on their right hand, while individual whatever has an extra toe on each foot. Individual, this one has 10 fingers and 10 toes. That's normal, y'all. Okay. These individuals show a range of incomplete penetrance. Right, it's not penetrance. Alright, okay. So this phenomenon of whether you act, because it's dominant, right? So let's start there. I was like, why did I pick this one? Okay, so let's start there. Okay, so it's a dominant trait, right? So why doesn't this person have extra fingers and toes? Because he's white, right? It means he's not affected. It means he has 10 fingers and 10 toes. That's what they're saying, right? Any one of these white blocks, right, has all the normal number, doesn't have any extra. Where anybody with a dark block has extra. Who cares at this point how many extra? Right? So this phenomenon, right, shouldn't these guys, right, it's a dominant trait. Shouldn't these two have extra fingers and toes? Oh, so you're saying this one's heterozygous, so that's why these guys don't have it? Okay, could be. Could be. Yeah, so that's probably the case right here, right? They're maybe heterozygous. Yeah, so, but if it's dominant, everybody should have it, right? If it's dominant, if you're heterozygous, you should have it, right?
Yeah.
Do you understand? Heterozygous doesn't matter. So the fact that one person has it means everybody should have it, right? It's dominant.
But it's being expressed differently, like it's either not being expressed at all or it's...
Right, this is what we mean by incomplete penetrance, right? This particular dominant trait doesn't, there's probably other things that factor in having extra fingers and toes. So just having this trait does not mean you're going to end up with extra fingers or toes. This is referred to as incomplete penetrance.
So
That's correct, but yeah, but what they're asking is another level to this. So whether you actually end up with extra fingers or toes, this is incomplete penetrance, because even though it's dominant, there's something that's stopping it from being expressed at that level of having extra fingers or toes. So now what they're saying, though, is this person has 6 fingers, right? So has 1 extra finger, basically, while this individual has an extra toe on each foot. Okay? So why is it this one only has an extra finger on their right hand, while this person has an extra toe and an extra toe on each foot? Why is it different? It's another term. Expressivity. They're referring to expressivity. How, okay, so the first step is it even penetrate, right? Do you even end up with extra fingers or toes? But then it can vary depending if you end up with extra fingers or toes as to how many and where.
But why is it referencing an individual who doesn't have any extras?
What does it say? These individuals show a range of incomplete penetrance, but they're talking more specifically, they're talking about the number and the location of the extra digits.
What's the question? Where's the question?
So would it be true if they're asking about?
So if they're saying, basically if they're saying this whole pedigree, right? Why doesn't everybody have extra fingers and toes? The answer would be because this is incomplete penetrance. But they're going one step further here. They're being more detailed. They're saying, why does one of them have only an extra finger and others end up with extra fingers or extra toes?
But where's the actual question? I don't even see the question.
So the statement, well, it's a statement because it's a true and false, right? It's actually a statement. It's not going to be a question. It's a false statement because the statement they're saying is why is this variation with as it relates to how many fingers or toes you have. I would be much more straightforward than this question. Remember how I said before I will give you the hardest possible question for homework and it will not be this complicated on the test. Not to mention I will be here, right? So if you're like, Miss Erica, what the hell? Right? I will say it again another way, hopefully, that is not like, what the hell are you talking about?
So if it said like these individuals show expressivity ranges of incomplete penetrance, would that be true?
Yes.
So it's missing the word expressivity,
Right.
and that's why it's false.
Yes.
Okay.
I just took the 9 out of 10.
Yeah.
You can take it again, yeah, but I was just like, all right. Yeah, I was like, I don't understand.
Yeah.
Would it be true
Uh-huh.
If that individual was heterozygous on the chart,
Mm-hmm.
then it would be an example, right? Then it would be true.
I don't know, this is a tough one, and very, yeah, let me put it this way. I wouldn't ask this one as true and false. I would ask it as a multiple choice. Which is this showing? Incomplete penetrance, incomplete, what are we talking about here? Incomplete penetrance, or are we talking about incomplete expressivity? Or, some other incompletes thrown in there as distractors. Does that make sense to you? All right. I have to agree that this is not very straightforward, because it is very nitpicky. And true and falses are like that, right? And I will put like a couple true and false, but they won't be as complicated as this. They will be straightforward statement. If the whole thing is true, it's true. But one little word, as you know, with true and false statements can make it false. And the one little word here is, it shouldn't say penetrance, it should say expressivity. Even though this is an example of penetrance as polydactyly in general is, but the specificity that they're talking about here of how many fingers and how many extra fingers or toes you have is an additional layer to penetrance, referred to as expressivity.
So
At what level is it expressed?
would complete expressivity be that they all have the same polydactyly, like we all have one extra pinky,
I guess so.
and then incomplete is like, I have an extra pinky, you have an extra toe. you
Yeah.
have a pinky.
So you have to give me an example of what's extra for it to be expressive.
Yeah. Yeah. If they just said that, they have extra fingers, they all have extra digits or extra fingers, and they didn't specify that they had different degrees of the expression. That's what they're showing you here. There's different degrees. One set has an extra finger where the other one has extra toes.
But then one has nothing.
Yeah, they're normal.
And
Because
that's
that's
just anything.
Yeah. I see what you're saying. I know. I'm done with this one, though. I'm done. My head's going to explode. Okay. All right. So I had a couple that I marked out of the homework for Chapter 5, since we haven't done any Chapter 5s yet. But they are in the homework. So I think I'll skip well, it's okay if they see it. I forgot to I don't want you guys to see the answer before I put it up there. But when in the recording, I don't care if they see. Me go look at the answer. Although then again, let me pause. Okay, so Chapter 5, at the end of the book, you know how end of the chapter they have questions. I'm going to go to concept question number 29. It's actually the last one of the concept questions. And it's an odd number, so it's not in the back of the book for you guys to look up. That's why I had to look at my answer key and copy it over. Duh. It was going, I just wasn't there. Okay, so we need to go to Chapter 5. Come on. And chapter. This is Chapter 5, right? No, it's Chapter 4. Because I was just like, those don't make sense. Crap. Did I copy the right ones? Yep, Chapter 4. Okay, sorry. It was 4. That's okay. I will pick some chapter 5s for you, though. Unless somebody has a chapter 5. No one's probably gotten that far. Chapter 5, I think, is relatively easy. I don't think. That one's going to hang you guys up. It's going to be 4 or the other ones that are going to get you. Okay, so chapter ending. Number 29. And then E1. I definitely want to go over. Okay, so. C29. So I could have put this in chapter 4. Oh, okay. All right. So 3 color coat patterns that occur in some breeds of horses are termed beige versus the chestnut brown and the, what is it, palomino, which is they're kind of brown with the golden tails. I looked up pictures. I should have put them into a PowerPoint. Most of you guys know what these horses look like? Palominos, I love them because it's better if you can see the pictures. Okay. They look like this. So their body is kind of brownish, but their hair, right, their mane and their tail is white. Where the beige ones, they're all like kind of whitish, like a beige color, right? The whole horse. And the chestnuts, they're all brown, right? Hair and tails. So what type of dominance pattern do you think this is? So if 2 palominos horses are mated, they produce 1/4 of the cream colored, 1/4 chestnut, and half of them a palomino. The beige ones, right, and the chestnut horses breed true, which means if you breed beige to beige, that's all you get. You breed chestnut to chestnut, that's all you get. So what pattern of inheritance do we have here? So what do you think the palomino is? You got it. It's incomplete. It's incomplete. It's the heterozygote, isn't it? And it's incomplete.
Okay, yeah.
Because chestnut is not being dominant, right?
Mm-hmm.
And beige is not being dominant. It's kind of, you could almost say it's a mix, yeah. But it's incomplete. Oh, y'all see that? Okay. Maybe so, it'll sink in. Okay. So then the next one that I wanted to do, where did I lost it? Okay, is this next example. Hopefully you guys can get this one right off the bat. If I could click on the right thing, I would be good. So this one's E1. This is probably the last one we'll have time for. Crap. So Mexican hairless dogs have little hair and a few teeth. When Mexican Mexican hairless is mated to another breed of dog, about half of the puppies are hairless. When 2 Mexican hairless dogs are mated to each other, about 1/3 of the surviving puppies have hair, and about 2/3 of the surviving puppies are hairless. However, about 2 out of the 8 puppies from this type of cross are born grossly deformed and do not survive. This pattern of inheritance is what? There's a key word here, right? Survival. So basically, if you get 2 defectives, what happens to you? You die. This is lethal genes, lethal inheritance. The whole survival clue is the clue there, right? Survival is the clue. And so remember with the minx cat, right? They end up with the shortened tail, but if you've got 2 defective alleles, you end up with a dead cat, right? Because their spine's so deformed, they can't survive. So number 3, you guys humor me for a second? Do you want to or no? If you have to run, you can. So number 3 is in sheep.
Wait, so what was the answer to the last one?
The last one is lethal. It's a lethal pattern. New slide. Paste. So in sheep, the formation of horns is a sex-influenced trait. The allele that results in horns is dominant in males and recessive in females. Females must be homozygous for the horned allele to have horns. So this is, as they already told you, it's sex influence, right? A horned ram is crossed with a pole, which is an unhorned ewe. The first offspring they produce was a horned ewe. I always have trouble saying that name. Female ram. What are the genotypes of the parents? Okay, so she has a horn, right? She is horned. So that means she has the dominant trait from what? Both parents. But her mom does not have horns, right? Females are, yeah, this one's a tough one. I'm just going to read the answer. If I can find it. The first offspring, the father's genotype is still ambiguous, right? Because he could be homozygous or heterozygous for the horned. The mother's genotype must be heterozygous because her phenotype is polled, which means that she doesn't have it, but she had to pass on that dominant one to her daughter in order for her daughter to have had it. But the dad, right, could be heterozygous and gave the dominant to the mom, or he could be homozygous with 2 dominant and gave, of course, only the dominant he had. Yeah, I know, you gotta kind of like think through it. Those are tough.
Do you have a piece of paper we can write on the test?
I'll let you write on the test, but I'll also give you guys extra paper too. I'm just going to save that in genetics folder.